Hello from Frankfurt. I'm very sorry that I cannot be with you in person, but I'm very glad to be invited and to present our recent data and discuss with you our efforts to disclose the mechanisms of cardiovascular aging. I will now share my screen and hope you can see my first slide, which is starting the presentation. And the second slide shows you that we are particularly focusing on the aging heart. As you know, aging is a major risk factor for cardiovascular disease and leads to a really high significant death and burden for society worldwide. Aging has many effects. On one side, it augments the vascular disease, leading to acute myocardial infarction and subsequent classical heart failure. In addition, aging alone can directly affect the heart, induce the so-called heart failure with preserved ejection fraction, which has a similar mortality and morbidity as the myocardial infarction-induced heart failure, but has a different pathomechanism involving the stiffness and increasing fibroblasts activation, but also an increase in endothelial cell dysfunction. We are particularly interested in the cellular signaling pathways which regulate cardiovascular aging, and in the past have particularly focused on the cardiomyocytes to understand the impact on cardiomyocyte aging, and also on the interplay between endothelial cells and neural cells in previous publication. In the study I would like to present you today, I would try to elucidate the effects of aging on the vasculature. Why this? As you know, the vascular cells and endothelial cells are most abundant cells of the heart. If you plate one endothelial cells from one human, you have this soccer field which you can pull. And these endothelial cells have important function, not only controlling oxygen and nutrient supply, but also control metabolism, export of weight. They control barrier functions and have a very important vascular niche function in aging. These are examples how endothelial cells can communicate in the heart, and you see here the arrows pointing to an intense interaction with cardiomyocytes, but also with other cell types in the normal heart. To gain insights into molecular changes of cardiac aging, we assessed the cardiac endothelial cell and other cell gene expression in aged mouse hearts, either taking young mice heart or 18-month-old mice heart. And this study was spearheaded by Julian Wagner, a postdoc in the laboratory. We assessed the gene expression in endothelial cells. You see here a single cell sequencing analysis. These are the endothelial cell clusters. And we made a very surprising observation, which puzzled us. We found that among the significantly upregulated genes in old heart endothelial cells, many GO terms associated with neuron death, neuron injury, axon injury, axon guidance, and so on. This was very surprising, and we asked the question whether this may mean that endothelial cells and neurons interact in the aging heart. Actually, it's well known since decades and hundreds of years that vessels and nerve align. These are very old drawings showing the vasculature and the nervous system. And also in the heart, you can appreciate here a very nice alignment and interaction of nerves with the vascular system. This is now a whole heart staining with 2G1, which is a neuronal marker. You see the cell bodies outside, and these projections are neurons, axons, which are invading into the left ventricle. If you look in depth in the histological analysis, you can appreciate that the nerves, these are again in green, in 2G1 stainings, align around the big vessels, like the smooth muscle cell positive pinkish areas, or the endothelial cells in the microcirculation. You can see here they follow also the lead in this direction, suggesting that possibly there might be a neurovascular unit in the heart, and we assessed whether there might be an effect of aging on this neuron-heart interaction. To do so, we first of all did cross-sectional staining. So these are the hearts now cut as cross-sections. These are young hearts, these are old hearts. And then again, we performed this 2G1 neuronal pan-neuronal stainings. And you can appreciate that in the left ventricle here, there is a diminished staining compared to the young left ventricle in this slide here. This is quantified here on the right-hand side, showing a significant decline at 16 months of age. This is about a 50 to 60-year-old human, so quite early during aging, there is a decline of axon density. These data suggest that aging reduces axon density in the heart, and particularly in the endocardium of the left ventricle. And the question is, what is the mechanism and subsequently then also the function? To assess potential mechanisms they may lead to the denervation of the aging mouse heart, we found genes associated with repulsive signals. These are signals which are known to reduce, or let's say, induce a regression of axons. And these are significantly upregulated in old heart endothelial cells compared to young endothelial cells. This is, for example, semaphorin 3A or netrin or SLIT3. These are all induced in the aging heart endothelial cells. Likewise, neuroprotective signals such as VEGF-B was declined in the aging mouse heart, suggesting that there is a disbalance of repulsive and neuroprotective signals. To gain first molecular insights, we focused on the repulsive signal, and here on semaphorin 3A, because we know that semaphorin 3A is known to affect the innervation pattern and also the heart rhythm, and also controls electrical remodeling and inhibits sympathetic axon growth. To do so, we first of all assessed whether semaphorin 3A is also controlled on protein level, and found that semaphorin 3A is indeed induced in the aging heart compared to the young heart. You see here the stainings on protein level. Moreover, adding recombinant semaphorin 3A to human iPS-derived cardiac organoids, which also contain nerves, induced a reduction of nerve density, as you can see here, demonstrating that also in vitro semaphorin 3A directly reduced nerve outgrowth. However, what is the effect in vivo? To test whether semaphorin 3A specifically being expressed in endothelial cells has an effect, we used an AAV vector which is targeted to endothelial cells in collaboration with Christian Kuppert using the system recently published. And you can appreciate this vector induces semaphorin 3A in endothelial cells, but not in non-endothelial cells of the injected mice. And indeed, if we deliver endothelial cell semaphorin 3A, this also induced a decline into G1 positive areas in these mouse hearts compared to the control vector. This suggests that overall denervation is a hallmark of aging, adding on top of the other known changes in the aging heart. There is an induction of repulsive signals associated with aging and a reduction of neuroprotective signals, which together reduce the innervation of the left ventricle. But what are the other upstream regulators? Of the hallmarks of aging, cellular senescence is particularly conserved and well established. Of course, it has a double-edged role because senescence is also a method to keep the cells in a non-proliferative state and prevent excessive overgrowth. But senescence leads also to the secretion of the so-called senescence-associated secretome, and these factors, which are secreted by senescent cells, have deleterious effects, particularly in aging. Interestingly, when we assessed endothelial cell senescence, we found that in the senescence endothelial cells, which are long-term passages compared to the young endothelial cells, they have also an induced semaphorin 3A expression, suggesting that possibly senescence or senescence-associated secretomes may play a role in reducing axon density. To test this hypothesis, we first of all looked into the time course of senescence and found that acidic beta-galactosidase was increased at 16 months old mice hearts, while axon density were declined at the same time, so coinciding. Moreover, if we took a replicative senescence model, which are telomerase deficient mice, also in this model, we could demonstrate a reduction of 2G1 positive areas indicative of reduction of nerves suggesting that the sole induction of premature senescence is sufficient to induce the denervation. But of course, this is in all cells of the heart. So the question is it sufficient to induce endothelial cell senescence? And to test this hypothesis is done by using a V-Catene progeria mice. This mouse has only an induction of endothelial cell senescence at early stages and was developed by Selma Osmanik-Myers from Vienna. And we found that induction of senescence in endothelial cells also specifically leads to a decline in axon density, as also shown here in these representative stainings. Overall, this suggests that in vivo senescence is associated with axon reduction of axon density. But what happens if we really mimic this in vitro? To test whether a paracrine effect is really mediating this effect, we used either young endothelial cells or long-term senescence endothelial cells. Then we used their supernatant, and this conditioned medium was then added to primary mouse cortical neurons. As you can appreciate, old endothelial cell supernatants reduce the density of nerves. You see here the axon density is declined, and this was reversed by neuropilin-1. This is a decoy blocking semaphorin-free activity, demonstrating that there is a rescue upon adding of this inhibitor onto the long-term senescence patches endothelial cell-induced reduction of neurostrontic activities. Together, this shows that denervation is associated with increased senescence, that induction of senescence reduces axon density, and that supernatants of senescent endothelial cells can reduce neural outgrowth in vitro. But can axon density be rescued by targeting senescence? So can we use a therapeutic approach to prevent this denervation? To test this effect, we used so-called senolytics. So senolytics are agents which specifically remove the senescent cells. There are several senolytics being developed, and we used a very first generation class of senolytics, which also have been tested in phase 1 clinical trials, so-called dasatinib and quercetin. And the combination of these senolytics was added in old mice for 2 months in comparison to the placebo-treated old mice or young placebo-treated mice. You can appreciate here that old mice have an induction of senescence and that this was prevented or reduced by senolytic treatment, as you can here see in the quantification. Likewise, coinciding, we could show that senolytic treatment rescues also the reduction of 2G positive areas in old mice, suggesting that indeed the prevention or inhibition of senescence is associated with a denervation of the aging mouse heart. What happens on the molecular level? We use single-cell sequencing again to test the effects on the repulsive and protective factors, and semaphorin-free, for example, was reduced by senolytic treatment, while VEGF-B expression was again augmented, showing that the senolytic treatment also ameliorated the profile of these neural-damaging molecules. What are the functional consequences of this denervation rescue? Of course, innervation has many functions, one of which is heart rate variability. This is declined with aging, so you can no longer respond to stressors, and reduced heart variability is associated with a poor prognosis. Denervation also can lead to silent arrhythmias, silent ischemia and lethal arrhythmias. In addition, repair and regeneration are impaired by reducing sympathetic or parasympathetic nerves. Thus, the autonomous control of the heart is important for regeneration. Finally, inflammation also controls inflammation and may play a role here as well for the aging of the heart, which is known to be associated with induction of inflammation. To test the effects of the senolytics, we used these different parameters. First of all, we assessed heart variability. You can appreciate here that senolytic treatment rescued the reduced heart variability of the old mice. And this also restored a very strong change in the day and night rhythms, which was impaired in the old see here in the red, in the day-night rhythm was really strikingly impaired in the old mice and was rescued by senolytic treatment. We found a reduction in arrhythmias in senolytic treated mice, a reduction in inflammation, as you can see here, and also a reduction in diastolic function, which is important for the overall heart function, as I have shown you in the initial slides. Together, our data suggest that senescence of vascular cells reduce the nerve density in the old mouse heart. This is in part mediated by semaphorin 3A, which leads to a regression of axons. If we treat old mice with senolytics, this reduces the number of senescent cells, leads to an increase in nerve density and normalization of heart ability, a reduced electrical instability, and an improved cardiac systolic function. If you would like to see more data, we have many more data, please have a look in this recent publication where you can find, as said, many details and more interesting insights. With this, I would like to conclude, put this a bit in a perspective. So it's also known that counteracting age-related VEGF signaling can promote health aging and extend lifespan, and we believe that counteracting vascular senescence may contribute to this effect. With this, I would like to thank the people who did the studies, many people of the group, particularly also, as I mentioned before, Julian Wagner, but also many collaborators who helped us providing mice and also helped us with electrophysiological assessment. And I thank you for your attention.